right. Hi everyone. I'm a PhD student at the Marine Biological Association in England, and today I want to introduce you to the wonderful world of coccolithophores and why we still need to consider using multiphasic approaches to study certain ecologically important clades. So, what are coccolithophores and why are they important? Some of you may have already seen the white cliffs that span much of the south coast of Northern Europe, which are in fact made up of calcium carbonate marine sediments from millions of years ago. And much of this calcium carbonate comes from coccolithophores. They're extremely diverse, unicellular microalgae, and they cover their cells in these elaborate, beautiful calcium carbonate plates. And we don't quite understand yet why they do this, but we think that it might be related to protection from grazers, sinking or light attenuation, but a lot of people think they might just do it because they're incredibly vain. So coccolithophores differ from other phytoplankton in that they don't only contribute to the organic carbon pump, but also to the inorganic carbon pump, because they don't only photosynthesize, but through calcification they release CO2 while the sinking of these calcite plates sequesters CO2 over time. So an area where coccolithophores are particularly abundant is the so-called Great Calcite Belt in the Southern Ocean, which you can see here by the calcium carbonate reflectance, which you can see from space actually. And we therefore went on a cruise to an area within the Great Calcite Belt in the Southern Ocean to describe coccolithophore communities. So we did this using scanning electron microscopy, which is a common tool to study coccolithophores to a high taxonomic resolution, and we also used metabarcoding of the entire plankton community to establish in which communities we find our coccolithophores. So we chose to use the same universal 18S eukaryotic gene markers that were used in the huge Tara Oceans expedition, so we can put our findings into a global context. And looking at the entire plankton community, we found a really diverse assemblage of clades, which was heavily dominated by dinoflagellates due to their high gene copy numbers. However, when we looked at the haptophytes, which should include our coccolithophores, we primarily found non-calcifying, non-coccolithophorid species. So we then had a closer look with the scanning electron microscope, and we found that there's in fact a huge diversity of coccolithophores in this area. However, most of these clades were not identifiable by metabarcoding, and I just want to briefly refer back to what I said earlier about the genetic markers we chose. Because one of the main reasons that we hardly find any coccolithophores in our data is that these general eukaryotic markers used in most metabarcoding studies of this kind have a mismatch in the DNA sequence of most coccolithophores. So despite them being such a ubiquitous part of the plankton, they're really, really underrepresented in even the largest ocean data sets like the Tara. So to address this mismatch, we resequenced our samples with markers specific to haptophytes to see if we get a better match with our scanning electron microscopy data. And while this did improve resolution, we still had a large portion of ASVs with no associated sequences in our data. And the reason is, of course, that we can't yet figure out how to culture most coccolithophore species, but getting a DNA reference sequence through single-cell sequencing would be a great step in the right direction. So part of our ongoing research is to try and determine which of these unknown clades could belong to the coccolithophore species we found using electron microscopy. So just to reiterate, coccolithophores are abundant and diverse in the Great Calcite Belt, but using general eukaryotic markers doesn't reveal this diversity. Using clade-specific markers can improve the resolution, but there's still many unknown clades, and we really want to highlight that metabarcoding alone can't yet replace multiphasic approaches for certain ecologically important clades. We still need to do microscopy and other methods to properly quantify the community, and we shouldn't solely rely on metabarcoding approaches alone. So yeah, I want to thank you for your attention, and all my collaborators, of course.